For those of you who don't already know, my name is Mr. Rodriguez, Mr. R for short. This is my email address, helpmemrr@gmail.com. Yes, I know, I was surprised it wasn't taken also. And then I said, well, I have the website, I might as well get the email, I might as well get the website also, helpmemrr.com. So you go there. You have any questions, you can email me, you go to the website. On my website, what I will be doing is setting up, or I have already rather, set up a practical review session. They have the PowerPoint that we're going over today already on there. On top of that, I have a practice practical. So you can go on there, test yourself out, and if you need the answers, just send me an email, all right? So, the way I like to start this whole thing off is kind of like my warning to you. Remember that your professor is number one. Your professor is number one. Whatever they say goes. I am secondary. I come in as just helping your knowledge of anatomy and physiology. I'll give you the ways that it helps me remember things. I'll give you tips and tricks that help you remember these things. As for anything else, the way that your professor states it, that's what you should be writing down, all right? Now, that being said, I have things that I will be going over that maybe some of you are not going over, and that's perfectly fine. If you want to put a blind eye to it, that's fine. Take a look at it. I mean, it's information, but you might not have to know that information. I know specifically for my class, in my lab class, I make them know more things because I add it on and I'm mean. Other people might not do that. They might be nice to you. But it's okay. I'm still going to go over everything. All right? Now, I'll start off by showing you how to get to the website and how to maneuver through that website. So what we'll do is you go to your web browser, whatever it is, be it Mozilla, if it's Internet Explorer. I know that I've seen a lot of problems with Internet Explorer. Mozilla actually has the best kind of, I guess, working of my website. So you see, this is my website. I'll hit the lights. Is that okay for the camera? We're good? All right. It's helpmemrr.com. This is the home page. I put a lot of blood, sweat, and tears into this website, so make sure you abuse it. You abuse this website. So you would go to Anatomy and Physiology Lab, click on it. You have two choices, AnP1, AnP2. When you'd go to AnP2, you have all the stuff for Anatomy and Physiology 2 as well. So you can go there also. But we are on Anatomy and Physiology 1. We have a little information about each lab. You click on the picture for each lab, and it brings you up to pictures about each lab. Now, we go to practical, click on the picture. You see there's two separate PowerPoints, the practical review PowerPoint and the actual practice practical. We will be starting off by going over this practical review PowerPoint. And then when I break you off into groups, you'll go over this practice practical yourself. This has all pictures. You can click on this little thing, make it bigger, and it'll bring you to something like this. And then you can go to Actions, save as a PowerPoint, print it out, do whatever you'd like to do with these particular slides. They're now yours, okay? Use and abuse it. So we'll start it off. Lab 1. Now, with Lab 1, the most important thing that we think about, is proper anatomical positioning. This right here. Feet about shoulder lengths apart. We have our hands onto our sides. We're looking upright. We're in the erect position like this. Now, everything goes back to proper anatomical positioning. Everything goes back to this anatomical positioning. Whether the patient's like this, however they are, moving around, everything has to go back to this anatomical positioning. That means this is my right-hand side. This is the patient's right-hand side. This is the patient's left-hand side. Every single time you're referring to left and right, every single time you're talking about something, it's the patient's right. the patient's left. So it says left or right, but it's all according to the patient. Okay, so this would be right-hand side, this would be left-hand side. We deal with body planes. With body planes, what we see, we have one that cuts like this. It separates superior and inferior. Superior and inferior, that's a transverse plane. Transverse plane. That can be up here, it can be over here, it can be over here, over here, over here. As long as it separates superior and inferior, it's considered a transverse plane. Mid-sagittal, right down the middle. Mid meaning middle, sagittal meaning up and down. It's going to separate right and left halves. Mid-sagittal means it's an equal separation between right and left halves. If I said parasagittal, that actually means just a separation between right and left, not directly in the middle. And then we have our last one, coronal or frontal plane. Go back to your definitions. One of your definitions for frontal meant forehead. So your forehead is anterior. This particular plane is going to separate the body into anterior and posterior sides. Anterior and posterior sides. So it'll be just like this. Front and back. I don't know what that front and back means. I kind of learn it for a second, and then I say anterior, posterior. Anterior, posterior. Ventral, dorsal. You use those terms. Now I can use that in several different ways. I could say, what is the name of the organ that is inferior to the diaphragm located in the right lumbar region? Woo! Woo! Inferior to the diaphragm. Diaphragm's right here. It separates the abdominal, pelvic, and thoracic cavity, right? So this is your diaphragm. I'm saying inferior, below it. It's in the right lumbar region. Right lumbar region looks like a bean. Kidney. Right kidney, yes. Be specific. Right kidney. Right? All right. So we take a look at this model. This particular model, we can ask so many questions. The reason why it's so nice is because I can go body planes, I can do organs, I can do the 9 regions, I can talk about anterior, posterior, I can talk about superior, inferior. I was going to say distal, proximal, but I don't think we can do that. I can probably find a different way to do that. But we can talk about so much on this particular model. So we'll start off with the macroscopic level, talk about organs, talk about structures found in this torso model. We have this guy right here made up of hyaline cartilage, goes from up, down into the lungs, the trachea. What you'll notice, this particular body is color coordinated. This light blue, you see it's the same light blue found here. That light blue is hyaline cartilage. Just go around that way. The light blue is the hyaline cartilage. So hyaline cartilage here, hyaline cartilage there. We see this dark red, this like burgundy color. That's going to be muscles. Muscles will be this dark burgundy color. We have this white color. That white color will be bones. This yellow down here, anytime you see yellow on a model, you're thinking nerves, especially torso models. And then we have this red, oh, what's this yellow thing over here? It's a nerve. We have a blue, we have red. How do we tell? Well, I have my little way to help you remember. Away. Red arteries carry blood away from the heart. Away. And if you think about like old superheroes, like Underdog used to go up and away, right? So I go, away. away. You have to have that like inflection in your voice to do it too. So away. Red arteries carry blood away from the heart. Red arteries carry blood away from the heart. The second one is BV2. Blue veins carry blood to the heart. Blue veins carry blood to the heart. BV2 sounds like a really bad boy band. It's like BV2, yo. Oh, oh, oh. Oh. All right, whatever. So BV2, blue veins to the heart. RA away, red arteries away from the heart. Away from the heart. Now, there are two pairs of examples that are exceptions to that rule. There are two pairs of examples that are exceptions, the pulmonary arteries and veins and the umbilical arteries and veins. Those are the two exceptions. But for the most part, when you see blue, we're going to think vein. Red, artery. Red, artery. Blue, vein. For the most part, that's going to be true. The only difference, like I said, those exceptions, those two pairs of exceptions, pulmonary arteries and veins, umbilical arteries and veins. And there'll be a different color. Arteries always carry blood away from the heart. Whether it's deoxygenated or oxygenated is basically said if it's either red or blue. This is your liver. This is your stomach. This is the transverse colon. No, it's okay. Think about the plane. Think about the plane. The plane separates superior, inferior, right? So this is transverse colon, ascending colon, descending colon. You have that S that makes the sigmoid colon. Think sigmoid S. You have the cecum, which is the first part of the large intestines. And the last part of the small intestines would be the ileum. How do you spell ileum? I-L-E-U-M. I-L-I-U-M refers to bone. Bone. I-L-E-U-M refers to intestines. I-L-I-U-M refers to bone. So the ileum connects to the cecum. The last part of the small intestines connects into the first part of the large intestines. You also, God bless you, also have a structure coming off of here. If you have lower right pains, the appendix, they say it might be appendicitis, right? It would actually be underneath this area. I don't have a picture, but I have the model over here that I will show right now. All right, so in this particular model, what we're seeing, it's a smaller model, but we still can see most of the things that are going on. We have a pair of lungs. We have a pair of lungs. Right lung, left lung. Very specific. Blue, veins, red, arteries. This light blue makes it cartilage. It's part of the windpipe, right? This is hyaline cartilage. Nice. Take out the lungs. We see these red and blue vessels, right? That's going to be your swap. Remember I said pulmonary artery, pulmonary vein. That's that exception. But you don't really go into that. So heart, right? This guy over here, liver, and underneath, gallbladder. Underneath would be the gallbladder. So gallbladder, right? And then this would be the liver. All right. Everybody's favorite. I know my favorite. My stomach. The reason why I love the stomach is because everything always refers to food and to movies for me. Food and movies. Stomach, I love food. The last part of the stomach actually enters into the first part of the small intestines. This first part of the small intestines, you see it on the posterior view. You see it on your posterior view. That's going to be your duodenum, spelt out D-U-O-D-E-N-U-M, like duodenum, but pronounced duodenum. That's your first. And what's this one? You said it before? Pancreas. the pancreas. Here we have transverse colon, we have ascending colon, descending colon. We have this sigmoid colon, and if we look directly on that ascending colon, this tiny little line coming off, it looks like a little tail, that's the appendix. And that appendix is located in the right iliac region. Right iliac region. We then have this guy over here. I guess I could take it out. The urinary bladder. The urinary bladder, made up of what type of tissue? What type of epithelial? Transitional epithelium. Nice. These are questions that could be asked. Remember, this is not just like lab 1, lab 2, lab 3. Everything has to come together. So this made up of transitional epithelium, located in the hypogastric region. Located in the hypogastric region. These guys, right and left kidneys, right and left kidneys. Red arteries, blue veins. What's this guy over here? The spleen. The spleen, located in the left hypochondriac region. So, let's go over that. Make our way to our whiteboard. We take our particular abdominopelvic region. Remember, we're talking about this region over here, below the thoracic region. Below the thoracic region. What separates the abdominopelvic and the thoracic region? The diaphragm. So we have these 9 body regions. The first one is above the stomach area. It's called the epigastric region. Think about this. Epi, above, gastric, stomach, right? So if we think about what kind of doctor we go see when we have stomach problems, we call it a gastroenterologist, right? Gastro is stomach. So above the stomach, we're seeing this epigastric region. Below the stomach, you will see hypogastric. Hypo, below. Hypo, below. Hypo, below. Hypo, below. Hypo, below. So you won't forget that. Hypogastric, below. Hypogastric. So that's below the gastric area. And then what is this thing that attaches us? Umbilical cord, right? So this would be our umbilical region. Now, everything is according to the patient. So this is right, this is left. So this is your right-hand side. I'm just making a little key. Left equals left-hand side, or L. So these guys over here, this is that kind of support that you get in your car. You know, it kind of pushes, it kind of feels like someone's punching you in the back because it doesn't really work. Lumbar support, right? If you have like a really high, expensive lumbar support, then it's going to work. But like in my car, it just feels like someone's like punching me. This would be right lumbar region, and this would be left lumbar region. Okay. How about over here when someone gets mad at you and they're on their iliac crest, right? How do we spell iliac again? I-L-I, right? E would be small intestines. So right and left iliac regions. And now this last one, you're going to look at me and say, what, you just lied to me. You said hypo, below, hypo, below. That's what you told me. But it should make sense now. You went over cartilage. You went over cartilage and connective tissue, right? What's the chondrocyte? It's a cartilage cell, right? Chondro is cartilage, cyte is cell. So this is the hypochondriac region, right? Hypo, below the cartilage. Our ribs are attached onto the sternum via hyaline cartilage called the costal cartilage. Below that cartilage are these two regions. Below the cartilage are the two regions, hypochondriac. And now this is not the same as hypochondria. When those of you going into nursing might seem that you will feel a little bit of hypochondria because you'll go over pathology and you'll go over diseases and how symptoms of diseases and signs, and you're going to say, do you feel thirsty? And you're like, I do. Do you go to the bathroom frequently? I do. Those are signs of diabetes. Just because you're thirsty and because you're like, oh man. So you'll see, you're like, maybe I have diabetes. You're going to go into that whole hypochondria, but that's something different. We're talking about below the cartilage, below the cartilage. Okay. Let's pull out those organs and see a different view. Let's see a different view. Let's see a different view. No? Doesn't want to work? There we go. Actually, this is just the same thing, just zoomed in. We have our liver, stomach, transverse colon, ascending colon, descending colon, jejunum, ileum, diaphragm. Right below the diaphragm over here would be the spleen, left lung, right lung, heart. How about if we pull that out? When we pull that out, what we see, stomach going into the duodenum, right? Kidney, adrenal gland. Kidney is in that right lumbar region because this is the right kidney.
Mr. R, if we're doing the website, is there a way for us to take the stuff off on the website? Not yet.
I wish I could do that. That would be cool. If I can get that, work on that. If I can get that, that would be cool. So heart, lungs, this whole area going from the trachea up to the larynx area. All right. Cool. Lab 3, the cell in the microscope. Now this particular lab, you should really take this as a gimme. It's just a structure and maybe a function, something easy that you just write in and say, okay, this is that, this is this, this is that. So the first one that we deal with is our favorite, the cell. Now with this cell, there are so many structures, but they are nice and easy to remember. I'll give you some tips on how to remember them. Now, what I like to do is pull this up. I'll erase these things over here, and now I can just draw on this picture. All right, so we'll start off with the big guy. This big guy, the brain of the cell, holds the genetic information. That is our nucleus. Our nucleus. Inside the nucleus, we have this orange ball called the nucleolus. Look at them. Yes, spelling does count, but this one, even more, it counts. I know personally I take off at, I don't know, like a quarter, half a point per spelling wrong. That I take off completely. Because I don't know which one you're talking about. Nucleolus, what does that do? Produces RNA. Nucleus houses the nucleolus, houses the DNA, houses nucleoplasm. That's a brain. This is only making the RNA. Yeah, it's big. It makes a big deal, but it is a major difference. Around this particular nucleus, we have an envelope. The nuclear envelope, it's a membrane. It works just like the cell membrane. It's a phospholipid bilayer. It's going to be semi-permeable. It works just like that. You have this, oops, plasma or cell membrane. It's as I was saying, it's a phospholipid bilayer. It's going to allow certain things in and out. It's for protection. We have this guy over here. The really big thing that we see is these shelf-like cristae that make up this mitochondria. Make up this organelle called mitochondria. You have one individual mitochondrion, multiple mitochondria. So this mitochondrion, what it's going to do is it goes through cellular respiration in order for us to make ATP. Now, how I always think about ATP, because they always make ATP look like this. ATP, right? It kind of looks like the old Batman shows, right? When you're like, bam, pow. So I was like, bam, ATP. So mitochondria goes through cellular respiration to create, bam. ATP. Wow, you guys are a tough crowd. All right. Now, we see this thing over here. I will give you a hint. Every time you see the suffix -osome, it means a sac filled with something. So this guy, the centrosome, is a sac filled with the centrioles. Now, once again, everything goes back to food and to movies. So these look like little Twizzlers to me. And the little Twizzlers are going to be your centrioles. Now I have this yellow jelly bean. Yellow jelly bean. Lysosome. A sac filled with lytic enzymes. See how that works? Lysosome. I like to think of the lysosome, think of it like Lysol. You know, kills 99.9% of bacteria, including H1N1 virus, right? That whole kind of big thing that they have, right? It kills off. Well, that's what this lysosome does. It's going to have digestive enzymes. So let's say this was a big phagocytotic cell. Let's say it was something like a white blood cell. As it went through phagocytosis, took it in, this particular lysosome is going to help it digest that material. Now, it's also known as suicide sacs because it can be harmful to the cell. It doesn't mean that it is harmful to the cell every single time. It can be harmful. What's this thing? Mitochondria. And this one? And that one? And that one? And that one? Doesn't matter what it looks like, as long as it's the same thing, it is, or it does matter. It is the same thing. It doesn't matter where it's located. It's the same exact look. Mitochondria, mitochondria, mitochondria. How about this guy over here? It really depends on your professor. They will either say peroxisome or vacuole. Either one, peroxisome or vacuole. But notice that's the same thing as this guy right up there. That's the same thing as this guy over here. Same exact thing. What's this guy? Lysosome, exact same thing. And this one? Okay, and this weird octopus looking thing. the Golgi apparatus or Golgi body. And now the job of that Golgi apparatus, the job of that Golgi body, what it does is it packages, it transports and receives information in the form of vesicles. You see these tiny things over here? Those are the vesicles. Those are the vesicles. This aqua-colored thing with the white dots on it, I'm so glad that you said endoplasmic reticulum and not ER. Because how do I know you're not talking about rough emergency room or something like that? Rough endoplasmic reticulum. And you're right, what is on there, those white things? Ribosomes. What do ribosomes do? I call them the boss. They're the boss of protein synthesis. They create proteins. So you have these rough endoplasmic reticulum with ribosomes on top of them. They actually look rough, so they say, oh, okay, rough endoplasmic reticulum. Smooth, we don't have a really good representation of smooth endoplasmic reticulum. And my friends, that is the cell. These things, like I said, this should be a gimme. You get there and you say, oh yeah, whoa, forgot about that one. What's this guy? Cytoplasm, or if you said cytosol, because technically that would be right because it is the fluid portion. You're not pointing to microtubules or intermediate filaments or microfilaments there, so you can't point to the cytoskeleton, so it's like the fluid portion.
And the darker blue would be what they're calling the smooth endoplasmic reticulum, or not really?
Like I said, there's really no good representation. If your professor is pointing to that for smooth endoplasmic reticulum, make sure that that's where you're pointing. Oh, and pinocytotic vesicle. Whatever your professor is pointing to, make sure that that's what you're using. All right. Now, next, the microscope. Yes, it's an older microscope, but it still does the job for us. This guy is the ocular lens or the eyepiece. Go down the body tube. You have your three objective lenses. The three objective lenses, you have scanning, low, and high. Scanning power is going to be 4x, low power is going to be 10x, and high power is going to be 40x. Now, this is called a compound light microscope. The reason why, you compound objective lens and ocular lens. Ocular lens is 10. So if you want to get total magnification, the entire magnification, you'd take 10 times whatever objective lens you're talking about. So 4 would be scanning. So 4 times 10, 40. Quick way to do that, you just add 0 at the end of that particular number, and that's multiplying by 10. So 4 times 10, 40. 10 times 10, add 0, 100. 40 times 10, add 0, 400. 400 times what the normal eye can see. That's what we're looking at. This guy. Now, when you go to a concert, right? Everything is happening on stage, right? It's on the stage. So this guy, if we're looking through this microscope, we're looking at the stage. On the stage, there's some clips that hold up your microscope slide. Microscope slide. It holds the microscope slide in place. There's these clips on the stage. What are they called? Stage clips. Go figure, right? Underneath the stage, there's a light. Underneath the stage, there's a light. Sub-stage light. Sub means below. So you think like sub-zero, below 0, right? Or submarine, below water. Sub-stage, below the stage, there's a light. Sub-stage light. These handles right here move the stage around. That's called the mechanical stage knobs. Mechanical stage knobs. That's the one that when you're looking underneath the microscope and when you're looking underneath, you want to move the slide left or right or up and down. That's what you're moving. Those are called mechanical stage knobs. Left, right, up, down. You have your coarse adjustment knob, your fine adjustment knob. You have your base, your arm. What's the proper way to hold a microscope? One hand underneath the base, one hand on the arm, like that. It's a proper way to hold it. Not like this, not like this. Perfect like that. What's this? Click, click. Power. It's the power button. On ours, it's actually behind here, that little flick, but this is like an old school microscope. So it's just like click, click. All right. Now everybody, you didn't get that on tape, did you? Nice. Can you get that for me? Mitosis, everybody's favorites. Thank you. Everybody's favorite, mitosis. Now, I'm going to show you slides. I'm going to show you the models. I'm going to show you how to remember certain things and how to just tips on how to identify each one of these slides. So the first thing that I start off with are actual slides. Now, what we'll see, once again, I'll take this, pull it upwards. I'm going to erase all this and have room to write. What you see, we have certain cells. With these cells, we're either going to have an intact nuclear envelope, interphase. Intact nuclear envelope, interphase. We'll have this kind of black lines right in the middle, just the chromosomes directly in the middle. We have prophase. With metaphase, we're going to have chromosomes lined up directly along the equatorial plane or metaphase plate. Anaphase, you might see a slight cleavage furrow, but the whole big thing is 2 lines of chromosomes. And then telophase, you'll see a bigger cleavage furrow. You'll see basically the production of 2 nuclei, telophase. A way to help you remember it in order, AnP, mat, or IPMAT. Interphase, prophase, metaphase, anaphase, telophase, IPMAT, IPMAT, if it helps. So interphase, we see this cell, and look what we see. Intact nuclear envelope. Intact nuclear envelope, that means interphase. Intact nuclear envelope, interphase. We have this cell with everything just floating in the middle, not along a line, but just in the middle. That's prophase. You'll see something like this. Chromosomes just in the middle. All right. What comes after prophase? Metaphase. So I took one from the distance that you're normally going to see it and one from a little closer. What's nice about this is you see chromosomes lined up in the middle. You also see these lines attaching on. Those are those microtubules. Those are those kinetochores attaching on. That's pretty cool. This is a really nice slide. You see again, metaphase. You see those things attaching on. Really nice slide. For the most part, you're going to see one single line of chromosomes. That will be your key. Look for one single line of chromosomes. Boom, boom. Whereas in anaphase, we will see 2 lines. You see how it's 2 lines of chromosomes, but they're in a different form. And then you have this slight indentation over here. Go around that way, go around that way. Slight indentation showing the cytokinesis occurring. Because towards the end of anaphase, we have cytokinesis occur. There's a chair right there. And then finally here, we have a very big indentation, even a line in between that cell. And we're creating 2 new nuclei. Telophase. Now, I know personally, I ask questions like, what form are these chromosomes in? Is it dyad? Is it a chromatid? Is it a chromatin? When we saw inside that cell model, the DNA inside that cell model is chromatin. So, how do we tell the difference? Well, there's dyad. That looks like this. That normal kind of chromosome look to it. That normal chromosome look to it. We have half of a dyad called a chromatid. Chromatid. And then we have that weird coiled looking thing called chromatin. Looks like that. Now, I have a way to remember this. Chromatin in interphase. So where are you going to find it? In interphase. Chromatin in interphase. Chromatin in interphase. Chromatin in interphase. So really the difference between chromatid and chromatin spelling-wise is 1 letter. So you're like, oh man, how am I supposed to remember that? Well, chromatin in interphase. On top of that, we said that a chromatid is half of a dyad, right? I think of the chromatid as like piece up, 8 town down, right? Piece up, 8 town down. Everything that I do has a meaning. Piece up, A town. So what phase is that? Anaphase. Oh, wow. You see that? A town down. Anaphase, right? So you have this chromatid, which basically looks like this, right? That's the dyad, the 2 chromatid. But if I separated them, you'd have 2 chromatid, right? Dyad, 2 chromatid. It's like I'm making a dance. Like, Anyway, so the way I remember that. You ready for this one? It has a double meaning also, okay? 2 of these make up 1 of these. All right. So we'll let that one sit in for a second. Marinate. So we said 2 of these make up 1 of these, right? 2 chromatid make up a dyad. Yeah, that makes sense. But the second meaning, how do you spell dyad with 2 D's? Oh, so notice how I said that. How do you tell the difference between the one with a D or an N? Well, 2 of these make up 1 of D's. So there's 2 D's in a dyad. The one with a D, chromatid, corresponds with the one dyad with a D. Dyad with a D. So now I can say dyad we're going to find in prophase and metaphase. Chromatid we're going to find in anaphase. And chromatin we'll find in interphase. And actually, when telophase is going from the chromatid back into interphase, it actually goes into chromatin also. So if I said a question like, what's the name of this phase of mitosis? Anaphase. What form of these DNA or chromosomes are? Chromatid. What are these? Those are the sister chromatid. All right. What if I said, what phase of mitosis is this? How do we know? intact nuclear envelope, intact nucleolus, and these tiny little squiggly things, chromatin in interphase. All right. Now I'm going to go onto my website and pull up a picture of mitosis. The picture of the models of mitosis. Look at that. And it's in order for you. Interphase, prophase, metaphase, anaphase, telophase, 2 daughter cells. So I'm going to zoom in. Okay, calm down. Okay, all right. We'll zoom in to look at each one. This is interphase, intact nuclear envelope, intact nucleolus. What are these things? Chromatin. All right, what are these 2 little black things? Centrioles. Now we move on. We go to early and late prophase. These guys are now in dyad form. Dyads. These centrioles are creating those spindle fibers and these little things. Asterase. Nice. So now you see the nucleolus and nucleus completely disappeared, right? You see the dyads kind of hanging around. Centrioles with these microtubules, with these spindle fibers trying to go and attach on. What's the next phase? Metaphase. We see in metaphase the dyads lined up along the equatorial plane or metaphase plate, right? We have these spindle fibers coming from our centriole, these asterase attaching on, asterase. We move on to the next phase. What's the next phase? Anaphase. Towards the end of anaphase, we start seeing cytokinesis occur, right? That's why we have this indentation, this cleavage.
So the blue indentation is the beginning of the cytophase.
Beginning of cytokinesis. Correct. So towards the end of anaphase, you have cytokinesis starting to occur. So that's why you have this indentation occur. So we have these, it was a dyad and now it's switched to chromatid with a D, to chromatid. Chromatid, 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 centrioles, spindle fibers, we see all that. And then we get to our 2 final stations, or our 2 final models. We see telophase, a prominent cleavage furrow, different than this one. This is just beginning. This is the actual cut. Think cleavage like a cut, like a cleaver, right? A cleaver is a knife that cuts, right? A furrow is a ditch. So there's a ditch where there's a cut, where this cell's about to cut. So that's what we're talking about. Now, you have this nucleus starting to reform. You have these going back into chromatin, where you have the 2 daughter cells in what stage? Nice. In interphase. You have the 2 daughter cells in interphase. It's called a life cycle of the cell. So it goes interphase, prophase, metaphase, anaphase, telophase, and then the 2 cells are in interphase who are just going to go through prophase, anaphase, anaphase, telophase. Keep going. Perfect.
So in telophase, don't we consider chromatin?
It's going back into chromatin. Going back into chromatin. Okay. Now, what everybody's been waiting for. I know it. Here's how I do this. I have a microscope here that I'm going to put actual slides on, and you'll tell me what they are. Okay, so now this is the part that everybody's been wanting to see. Now, like I said, I have slides. I'm putting them up as we speak. This is just a slide that I took from a slide tray. It's really what I did. We're looking at epithelial tissue. Now, unfortunately, on the practical, it's not going to say what type of epithelial tissue this is. It's just going to say what type of tissue. But I can teach you some ways that will help you remember how to look or distinguish between epithelial tissue. Now with epithelial tissue, this is how it goes. You will always see some sort of basement membrane and apical end every single time. Unless we're talking about this guy over here, because then it's a little difficult. But everything else, we'll see it. Now this is simple squamous epithelium. Simple squamous epithelium. Now, what you see, it looks pretty much like a single layer of these weird shaped cells. The cells look like fried eggs, so it has that irregular border, right? One single line of fried eggs looking like this, it's spaced out. That's simple squamous epithelium. We find this in the lungs. In the lungs. Now, if you look at individual cells, you see it looks like a weird border with a nucleus in the middle. Weird border with nucleus in the middle. I would take a look at all of the slides together and just distinguish. This one's very, 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 like, it pops out. You may look at this one and look at adipose connective tissue and say, what's the difference? Well, adipose has really thin lines, no nuclei. Or if you'll see a nuclei, it's just one over on the side. This, you see many nuclei. Many nuclei. All right? Well, let's give you a stumper. You know what? I'm actually going to change the way I do this. I'm going to go over all the tissues and then throw on slides and have you think about them. I think that one will work better. So let's go over which one's that again? This is going to be simple squamous epithelium. You see multiple nuclei. It does look like that adipose a little, but you have a whole bunch of nuclei. Okay? So when you see the many nuclei around, you're thinking to yourself, simple squamous epithelium. Now, notice how I'm saying it. Simple squamous epithelium. I know personally, I am a stickler. If you do not say simple squamous epithelium, I take off points. In fact, the whole thing is wrong because simple squamous epithelium, you're telling me how many layers, you're telling me the shape of the cell, and you're telling me that you know what type of tissue it is. That tells me you know what you're talking about. If you just said squamous, I'm like, squamous what? Squamous epithelium. Squamous epithelium what? Simple squamous, stratified squamous. How do I know you don't think it's pseudostratified when we know it's not? So we're making sure, I know personally that's how I say it. Get used to saying the whole thing and you have no questions about getting it right or wrong. How about this one? Ooh, this is a beautiful slide. Simple cuboidal epithelium. Remember I said it always deals with a basement membrane and the apical end or free surface. Once again, going back to food, looks like donuts. Looks like donuts to me. The hole in the middle, or even a bagel. Have the outside with the hole in the middle. These cells are box shapes. They look like cubes, cuboidal. Now, even though it looks like multiple tissues on top of each other, notice that it's one single line. Single line. That single line makes it simple. Simple cuboidal epithelium. Simple cuboidal epithelium. What's these little circles? The nucleus. How about this one? Simple columnar epithelium. Now it's kind of close up, but you do see the fact that this would be basement membrane and the nuclei point towards the basement membrane. This would be the apical end or free surface. And what's this guy?
Goblet cell.
Goblet cell secretes mucus.
What was
Name of the tissue?
no. And the other one? You said the first one was lungs. What was the second one?
Oh, either thyroid or kidney. And then this one's going to be in the intestines. How about this one? Stratified squamous epithelium. you're going to look for this coastline. It kind of looks like if water hit the sand and you create that line in the sand. This area right over here. This is what you're looking for. And then on top of that, this is truly the only tissue that looks like it has a whole bunch of cells on top of each other. Look how it looks like a whole bunch of cells on top. It's not like one single row. It's a whole bunch of them. Apical end free surface, basement membrane. Each individual would be a nucleus. And then it is a stratified squamous epithelium. Here's a little hint. Do we have stratified cuboidal? No. We have it in our body, yes. We have stratified cuboidal epithelium in our body, but we don't have a slide. Maybe that'll help. Do we have stratified columnar epithelium? Yeah, we have it in our body, but do we have a slide? So truly, there's two stratified tissues that we deal with. One is transitional, and one is stratified squamous epithelium. So if you see a whole bunch of this, you have stratified squamous epithelium.
Located where?
Skin, scalp.
So even though we don't have a slice for those, are you still
Me?
yes, you?
I'll be nice. What's this one? This is transitional epithelium. How do we tell? We look at the starfish. See this star? That's going to be our transitional epithelium. Now, apical end, free surface, basement membrane. This is stratified. It is stratified.
So would that be stratified transitional? Or just
No, you can just say transitional epithelium. Now, with this particular, you're going to find it in the urinary bladder, in the ureters. The function is to be able to stretch without rupture. You know, when you have to like really, really go to the bathroom and you're like, ugh, doing your little pee-pee dance, right? The fact is that our bladder won't just and blow up. It's going to be able to stretch without rupture. Okay, let's move on to connective tissue. I'm going to go through all the tissues and then I'm going to test you. I think that's good. It's a good way to do it. What's this? Adipose connective tissue. It looks like a whole bunch of popcorn. Doesn't it look like popcorn? Like 1, 2, 3, like a kernel. Marshmallows, all right. Whatever. This is very, very prominent. You look at this and you see it and you're like, oh, that's fat. That's adipose connective tissue. Each individual would be an adipocyte. Adipocyte. Okay, how about this one? Areolar connective tissue, or loose areolar connective tissue. You see a macrophage, you see fibroblasts, fibers. How many fibers are going to be on this? Lots and lots, all of them. Collagenous, reticular, and elastic fibers. All right. How about this one? This one's nice and easy too. Blood connective tissue. Because we see a whole bunch of red blood cells known as erythrocytes. Erythrocytes. If you don't have to say erythrocyte, then okay, fine. But I know I'm preferring erythrocyte. And then these are white blood cells, leukocytes. Leukocytes. This one's nice and easy. In between, this white in between is going to be the plasma. Plasma. Okay. What about this one? That like punches you in the face when you see that. You see the whole bone. It looks like if you took a tree, chopped it, and you have those rings, those tree rings. Now the thing about this particular connective tissue, we actually can ask a lot of information. Right here, Haversian canal. We have the osteocytes. We have the lamella. We have the canaliculi. The canaliculi. This is osseous connective tissue or bone connective tissue. This one comes right out at you on the slide. but you still have a lot of structures you have to go over. Haversian canal. How about this one? White fibrous connective tissue. If they said dense regular connective tissue, either one is correct. It is both the same thing. What you're seeing is a wave. Now I know for Professor Kingsley, she's going to turn it. She's going to have up and down. But I'm not as nice. I have it either way. So it has the waves. If you see these waves, you're thinking dense regular or white fibrous connective tissue. What you have here is a whole bunch of collagenous fibers all packed together and kind of putting these fibroblasts, kind of like making them smaller. So you have these tiny fibroblasts with these collagenous fibers kind of compacting them. When you see waves, think white fibrous connective tissue or dense regular. When you see tree rings, you're thinking bone. When you're seeing popcorn, adipose connective tissue. When you look into the microscope and it looks like eyes are looking right back at you, hyaline cartilage. You see these guys? They're looking directly right over there. It's like, hey, saying what's up.
They're usually here.
Correct. Boom, 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 boom. Eyes looking directly back at you. So you look inside, you see these eyes looking back at you. These are chondrocytes in lacuna. Chondrocytes in the lacuna. Hyaline cartilage, where do you find it? Nose. Trachea. Costal cartilage. Oh, nice. Articulations of long bones. And what's the other one? Oh, don't cheat. Embryonic skeleton is made up of hyaline cartilage. How about this one? Scary eyes. They look like eyes, but they're scary. Elastic. It looks like someone should be like, ah, right next to it. You have these same eyes looking back at you, but it has like a Halloween feel to it. These black lines coming off, those are the elastic fibers. These guys are the actual chondrocytes in lacuna. Where do you find elastic cartilage?
Outer ears.
Pinna of the ear, right? Outer ears, the epiglottis. What's this down here?
Adipose.
Nice. Adipose connective tissue. I can't fool you guys. All right. How about this one? Fibrocartilage. You see the chondrocyte in lacuna, and you see these thick collagenous fibers. Collagenous fibers there for tensile strength. It's strong. So these particular, this particular cartilage, you're going to find intervertebral discs, the symphysis pubis, anything that's going to have to be able to withstand tension. Okay. What's this called? The structural unit. There you go. Just want to let everybody know that's my student. Just saying. No, I'm kidding. Yes, it is an osteon, the structural unit of a bone. Structural unit of a bone is the osteon. So make sure you know that. That is important. So if they were saying something about the osteon model, this is what they're talking about. And when we looked at underneath the microscope, when we saw those tree rings, right? This is what we looked at. This is just a model of what we looked at underneath the microscope. So you see the Haversian canal. You see the osteocytes. You see the canaliculi and the lamella. Also, you see inside that Haversian canal, blue again was vein, are arteries. So you see a vein and an artery and nerves coming through. In order for us to make up a bone, you actually have all of these, a whole bunch of these together. A whole bunch of these together. And you make up that whole entire bone. That's why when we looked at it underneath the microscope, That's what we were looking at. This whole osteon. This is an osteon. All right. How about that? I hear both. Here's how you tell. Here's how you tell. Look at the nucleus. It's on the outside. If the nucleus is on the outside, if it's on the peripherals, it's going to be skeletal muscle tissue. You'll see these nuclei on the peripherals, and you see these striations. The striations, skeletal muscle tissue. If the nuclei are on the peripherals of this long kind of fiber, then you're going to see that it's skeletal muscle. If you see branching, you see how they have white lines in between? This is kind of branching off. That's cardiac muscle tissue. You have these faint striations, but you see these darker lines called intercalated discs. Intercalated discs. So branching within cardiac muscle tissue. Up and down with these striations and the nuclei on the outside, skeletal muscle tissue. Where's the nuclei located? Pretty central here, right? See, it's not on the outside like on that skeletal muscle tissue. And then finally, we have like a paintbrush, right? We take a paintbrush. Ow. That hurt. We take a paintbrush and we paint across this particular picture. We have a smooth kind of look to it, smooth muscle tissue, also known as visceral muscle tissue. Now that word, visceral, actually keys off to something else. Covers the organs. So we think about serosa, right? Serosa that covers the body, right? That membrane. We have visceral or parietal. Parietal, parent provides the home. That's how I like to think about it. The parent provides the home. What's the home of an organ? The cavity in which it stays. So the parent provides the home, parietal. That serosa, parietal, covers the cavity. Visceral covers the organ itself. So if we said something like, what's the name of the serosa that covers this particular organ? Visceral pleura. Or what covers the cavity? Parietal pleura. What's the name of the serosa that covers this organ? Visceral pericardium. Nice. Yes. Awesome. Visceral pericardium. All right. So we'll go over the skin model in a second. What I want to do is now go over those different slides. I'll put up a slide. You tell me what it is. Now I can just go random. We're just going to go random. This is simulating your practical, something that you'll see in your practical. Give it a second, hold on. Whoa. What's the name of this tissue? Nice. We see a column, single line, basement membrane, apical end or free surface. It looks like one column. Simple columnar epithelium. As a hint, for simple columnar epithelium, the nuclei will always point towards the basement membrane. So you see nuclei pointing on this direction, so that's where the basement membrane is. Basement membrane. All right, so you guys can't be fooled, huh? Let's see. About Oh, man. Trying to be lazy. Hold on. Skeletal muscle tissue. Why are we saying it? Striations gives up number 1, and number 2, the nuclei are on the peripherals, on the outside. Since they're on the outside, they actually key off to us being skeletal muscle tissue. All right. So you guys are good, huh? Oh no. This is to appease everybody. Come on, you guys should get this. White fibrous connective tissue. The reason why you see these waves and these flattened fibroblasts. All right. Hello. Ooh. All right, nice. Hyaline cartilage. We have hyaline cartilage. No. Hyaline cartilage. So we see that they're the chondrocyte in lacuna, right? How about, whoops. Come on, leg. Ooh.
Pseudostratified ciliated epithelium.
All right. Pseudostratified columnar epithelium. How do we tell? Cilia. Cilia is how we tell. It kind of looks like it's stratified, but it doesn't. It has these kind of weird column shapes to it. All right. I thought I can get you. You should get this one in like 2 seconds. All right, give me a second. Let me at least, before you, I know, before you say it, hold on. Let me set it up. Oh, man. Really, guy? Bone connective tissue, right? I gotta have people at home who's watching that they still gotta see it too. Bone connective tissue, right? We have our Haversian canal, lamella. We have all the things that we talked about. This one should really like just hit you right as soon as you look at it. You're like, oh, okay, good. I know what this is. How about this one? Stratified squamous epithelium. We see a whole bunch of cells flattened, right? And it looks like multiple layers. You also see that coastline that I was telling you about. See the coastline look like a whole bunch of cells flatten together. All right. How about talk about perfect. Hello.
Oh, it's the paintbrush one. Yeah, smooth muscle.
Smooth muscle. Smooth muscle tissue. Looks smooth like the paintbrush, bigger nuclei, right? It's the paintbrush one. You know, those things are going to help you remember it, to tell you the truth. It will help you. Yep, yeah. Too close. Cardiac muscle tissue. How do we know? Look at the branching. Nuclei right in the middle, and these intercalated discs. The intercalated discs. Loose areolar connective tissue. What's this big guy over here and over here? Macrophage. All right. All right, I can't trick you guys. How about if I get, not really trick, I can't stump you guys. That's really what I'm trying to do. That was a little too easy. Blood connective tissue. You see red blood cells, erythrocytes, white blood cells, leukocytes.
Did you ask plasma? Did you ask in the middle?
Say it again? Plasma?
Plasma.
Yeah, of course. That's a good question. All right, let's make this. Let's see if this one works out for us. I'll try to find something here. Look at that. I tricked it at you. I got it. Take a look at this one.
Daughter cells.
Telophase. The two daughter cells are starting to be made. What about this one over here? Interphase. How about this one over here? No, not that one. That one doesn't show it too well. I don't like it. A lot of interphase. Why does it make sense that there's a lot of interphase? Very nice. That's where they spend most of their time. This is really hard to navigate on this thing. All right, well, if I find another good one, I'll send it your way. So now what I want you to do is pair up with somebody, get to one of these computers, and go onto my website and take the practice practical. On this practice practical, what you're going to be able to do is see different structures. You're pointing to different structures. You could stay right here. And I'll be running around answering questions. This model will be out here. Oh, actually, I'm sorry. One more thing. Skin. One more thing. All right. This is the model of the skin. Okay? Model of the skin. We have this first layer, which is on top. Epi means above. So we have the epidermis. We have directly underneath the epidermis, we have our dermis. And technically under here, this would be all hypo, below, hypo, below. So this would be hypodermis. What's the first layer or the outside layer of the epidermis? Stratum, starts with a C, ends in an ornium. Corneum, stratum corneum, good. We then have these projections coming out, or no, I lied, that hair coming out. That's the hair shaft, and the projections coming from the dermis are called dermal papillae. Now what we see, if I ask the question, what's the name of the tissue that comprises the epidermis? Stratified squamous epithelium. Stratified squamous epithelium. Avascular or vascular? Avascular. Avascular. Notice all the capillary loops are inside this dermal papilla, not in that epidermis. It is avascular. Next thing is pointing to the Meissner's corpuscle. It's a light touch receptor. Then you have these red and blue things. What are they called? Veins and arteries, but what are they called? Like its general name? Blood vessels. We have this gland on our hair that secretes sebum or oil. Sebaceous gland. Sebum is oil. And then we have the muscle that pulls on the hair in order to give us goosebumps, increased surface area. What kind of muscle do you think that is? Do we go, hair stand up now? No, it's involuntary. What are the two types of involuntary muscles? Cardiac and smooth. Is that inside the heart? So it has to be smooth muscle. So we have the erector pili muscle. We have the sweat gland or sudoriferous gland. We have the hair follicle itself. And then we have this yellow stuff underneath. What is that? Adipose connective tissue. So that's usually located subcutaneously, below the skin. Hypo, below. And then we have our Pacinian corpuscle, which is for deep pressure. If we're talking about feeling deeply, that's for deep pressure, Pacinian corpuscle. Now you guys can go over there and team up. Thank you very much.